Dear learners, I am Ramya, working as Assistant Professor in the Department of Biotechnology, St. Joseph's College of Arts and Science for Women, Hosur. Today, we are going to see the topic Structure and Functions of Cyanobacteria. What is cyanobacteria? Cyanobacteria is a blue-green algae. It is called as prokaryotes with cell envelopes typical for gram-negative bacteria. The cell envelope consists of four distinct layers: the plasma membrane, peptidoglycan layer, the outer membrane, and in some cases the surface or S layer. Like bacteria, the cell of cyanobacteria also consists of mucilaginous layer called sheath, the cell wall, plasma membrane, and cytoplasm. Sheath. Usually, the cell of cytobacteria are covered by hygroscopic mucilaginous sheath, which provides protection to cell from unfavorable conditions and keep the cell in moisture condition. It has thickness, consistency, and nature of sheath are influenced by environmental conditions. Cell wall. After observing the cyanobacterial cell under electron microscope, it appears multilayered present between the sheath and plasma membrane. The cell wall consists of four layers designated as L1, L2, L3 and L4. The layers L1 and L3 are electron transparent, whereas L2 and L4 are electron dense. The L1 is the innermost layer of the cell wall present next to the plasma membrane and a thickness of about 3 to 10 nanometer. L2 is a thin electron dense layer which is made up of glucosamine, alanine, glutamic acid. The layer L2 provides shape and mechanical strength which has a thickness of 10 to 1000 nanometer. L3 is again electron transparent layer of thickness 3 to 10 nanometer. The outermost layer is L4 which is thick and electron dense layer. It is appearing wrinkled and undulating. All the layers are interconnected by plasmodesmata. Numerous pores are present on the cell which acts as a passage for secretion of mucilage by the cell. Next is about plasma membrane. The cell wall is followed by bilayer membrane called plasma membrane or plasma lemma. It is 70 Armstrong thick, selectively permeable and maintains physiological integrity of a cell. Plasma membrane sometimes invaginates locally and fuses with the photosynthetic lamellae called thylakoids, which forms a structure called lamellosomes. The plasma membrane encloses cytoplasm and other inclusions. About cytoplasm. Cytoplasm is distinguished into two regions, the outer peripheral region called as chromoplasm and central colorless region called centroplasm. Chromoplasm contains vesicular structures flattened and called photosynthetic lamellae or thylakoids. Thylakoids may be peripheral, parallel or central. Besides photosynthesis, thylakoids have capacity of photophosphorylation, Hill reaction and respiration process. Depending on the physiological conditions, it is arranged, and several photosynthetic pigments such as chlorophyll A, chlorophyll C, xanthophylls, and carotenoids are present inside the lamellae. On its upper surface, phycobilisomes, called as billiproteins of about 40 nanometer, are anchored by a protein. Phycobilisomes comprises of three pigments: phycocyanin C, phycocyanin, and phycoerythrin C. These three pigments harnesses lights in the sequences. Next is about centroplasm. The centroplasm is colorless and regarded as primitive nucleus devoid of bilayered nuclear membrane and nucleolus. Several grains that can take strain as dispersed in cytoplasm. This is the diagram of cyanobacterium, which has DNA fibril in the center and protein body, and lamellosomes, thylakoids present at the side, lipid granules and cyanophysian granules, gas vacuoles are present that has covered by a mucilaginous sheath, and phosphogranules present inside the cell wall. Cytoplasmic inclusions. Several glycogen granules, oil droplets, and other inclusions are dispersed in chromoplasm as well as in centroplasm regions. Cyanophysin. The cyanobacteria accumulates nitrogenous reserve material called cyanophysin or cyanophysian granules. When grown at conditions of surplus nitrogens. They can be observed under light microscope as they accept neutral red or carmine. Next is about gas vacuoles. In many cyanobacteria, example Anabina, Gliotrichia, Microcystis, Oscillatoria, etc., the gas vesicles of viscopseudovacuoles of different dimensions are found. The cytoplasm lacks vacuoles. The vesicles are hollow, rigid, and elongated cylinders, 75 nanometer in diameter and 200 to 1000 nanometer long, which is covered by 2 nanometer thick protein boundary. The end of vacuoles are conical. The functions of gas vesicles is to maintain buoyancy so that the cell can remain at certain depth of water where they can get sufficient light, oxygen and nutrients. Floating and sinking phenomenon is a key feature found in free-floating cyanobacteria. Carboxysomes. Carboxysomes are polyhedral bodies containing 1,5-ribulose biphosphate carboxylase called as rubisco. Next is about DNA matrix. Like other prokaryotes, the cyanobacteria also contains naked DNA fibrils dispersed in the cytoplasm. DNA materials lacks nucleoplasm and like E. coli contains a histone-like protein that binds with DNA. The total number of genomes is not yet known, but in Agmenellum 2 to 3 genomes have been reported. Like eubacteria, cyanobacteria also has 70s ribosomes. Similar to bacteria, cyanobacteria contains covalently closed non-functional circular plasmid DNA. Some specialized structures present in cytobacteria. They are hormogones, exopores, endospores, nanocysts, heterocysts, exospores, endospores, actinides, etc., which are produced in cyanobacteria. Hormogones and hormocysts. Hormogones are the short segments of trichomes produced in all filamentous cyanobacteria. It is produced by several methods such as fragmentation and separation methods. The hormogones shows gliding movements. Each hormogone may develop into a new individual. Endospores, exospores and nanocysts. The non-filamentous cyanobacteria generally produces endospores, exospores and nanocysts. They are produced inside the cell. During endospore formation, the cytoplasm of a cell is cleaved into several bits which are converted into endospores. Some of the cyanobacteria reproduces by budding. The spores produced through this method are called as exospores. Next is about actinides. The different groups of cyanobacteria are capable to develop vegetative cells into spherical perennating structures called actinides or spores such as nostoc, rivularia, gliotrichia, etc., which we already saw. During unfavorable conditions, the vegetative cells accumulate much food, enlarge and become thick-walled. These are formed singly or in chains. Actinides possess cyanophycean granules, hence these appears brown in color. Heterocyst. Heterocysts are the modified vegetative cells. Depending on nitrogen concentration in the environment, heterocyst formation occurs. During differentiation, several morphological, physiological, biochemical and genetical modification takes place only in heterocysts. They are slightly enlarged cells, pale yellow in color, containing an additional outer investment. These are found mostly in Oscilloteraceae, Rivularaceae, Nostocaceae and Cytonemataceae. What is the other name for cyanobacteria? This is the question for today's learning. Thank you.